What are plastids? They are photosynthetic organelles found in eukaryotic cell like the plant cell but absent in animal cell. When we differentiate a prokaryotic cell from a eukaryotic cell, one very important point is the presence or absence of ribosomes. Hello everyone, welcome back to session 4 of this interesting chapter called Cell, the Unit of Life. Hope you remember in the last session we studied about the endomembrane system, about the Golgi bodies, rough endoplasmic reticulum, lysosomes, right? So what are we going to study in today's session? The continuing the concepts of the remaining concepts of this chapter called Cell, the Unit of Life, that is about the plastids. The coloring pigments. Hope you remember, like when we differentiated a plant cell from an animal cell, absence of vacuole, absence of plastids in case of an animal cell, and presence of vacuole and presence of plastid in case of a plant cell. Here you can see the structure of a plastid, that is the chloroplast. You can see it is a double membrane structure and having thylakoid, and the inner content of the chloroplast is called stroma, and here you can see the coin-like structures which is piled up one above the other. They are called thylakoids, and this unit is called as grana, and the inner part is called stroma. Right, now let us understand what are these plastids. They are very, very important photosynthetic pigments. You can call them as the photosynthetic organelles found in eukaryotic cell like the plant cell. So what are plastids? They are photosynthetic organelles found in eukaryotic cell like the plant cell but absent in animal cell. And plastids are found in all plant cells and in some euglenoids. Right, and they bear some specific pigment, impart specific color to the plants. I think all of us know most plants are green in color, and that green color is due to the presence of the green coloring pigment called chloroplast. Right, so they bear some specific pigment, impart specific color to the plants. Based on the type of pigments, plastids can be broadly classified into chloroplasts, and I hope you remember there are different colored flowers with a different color, blue color, yellow color, red color, carotene, right? So they are called as chromoplasts. Colored pigments are called chromoplasts. Green color pigments are called chloroplasts, and colored pigments are green pigment is called chloroplasts, and colored pigments are called chromoplasts, and colorless pigments, they are called as colorless leucoplasts. So here you can see the structure of the chloroplast. This is the structure of a chloroplast. So what are plastids? They are photosynthetic organelles found in eukaryotic cell. Plastids are found in all plant cells and in some euglenoid. They bear some specific pigment which is going to impart that specific color to the plants. It might be chloroplast, it might be chromoplast, right? Like xanthophyll, phycocyanin, then fucocyanin, then we have this carotene, right? Based on the type of the pigment present, they are going to impart specific color to the plants. So based on the type of pigment, plastids can be classified into 3 main types. What are they? Chloroplasts, chromoplasts, and leucoplasts. What are chloroplasts? The green coloring pigments are called chloroplasts. What are chromo? Chromo means what? Colored. So the colored pigments are called chromoplasts, and leuco, colorless. They are called colorless pigments. Now, the chloroplasts, as we know, contain chlorophyll. I think all of us know plants are autotrophic. They synthesize their own food through a very important process called photosynthesis. And what are the raw materials for photosynthesis? Carbon dioxide, water, sunlight, and very, very important is chlorophyll. So the chloroplasts contain chlorophyll and carotenoid pigments and trap what? Solar energy. Why chlorophyll is so very important for photosynthesis for the plants? The chlorophyll A and chlorophyll B. Chlorophyll A is the main photosynthetic pigment which is going to trap the solar energy. I think all of us know the prepared food of plants is called starch. Starch is nothing but it is in the form of chemical energy. Chemical energy. So the plants utilize the solar energy, convert it into chemical energy, and when we have this as a form of food, we get it is converted into kinetic energy. So solar energy into chemical energy into kinetic energy. So the chloroplasts contain chlorophyll, and this chlorophyll is going to trap the solar energy for photosynthesis. So like for example, we come across the chromoplasts. Fat soluble carotenoid pigments like carotene, xanthophylls are present. The leucoplasts, as I said, they are colorless plastids of varied shapes and size with sorted nutrients. They are colorless plastids, but they have sorted nutrients. Like for example, we have 3 types. This is very important, which is also asked in your competitive exams. Amyloplast stores carbohydrates. For example, like starch in potato. Then elaioplast store oils and fats. Aleuroplast store proteins and minerals. So they are all what? Colorless plastids. They are colorless plastids. Hence the name leucoplasts. They are all colorless plastids. I repeat, the chloroplasts contain chlorophyll and they trap the solar energy, which is very much required for photosynthesis. So when they trap solar energy, an electron is expelled and that electron passes on through a series of electron acceptors. So in the chromoplast, fat soluble carotenoid pigments like carotene, xanthophyll are present, and the leucoplasts are colorless plastids of varied shapes and size with sorted nutrients like amyloplasts store what? Carbohydrates. Amyloplasts store carbohydrates. Elaioplasts store oils and fats, and aleuroplasts store proteins and minerals. Chloroplasts are found where usually we come across the chloroplasts found in the mesophyll of a leaf. As we know, leaf is the main photosynthetic apparatus of a plant body. So how are these chloroplasts? As you have already seen the structure of the chloroplasts, these are oval, spherical, discoid and even ribbon-like organelles. So chloroplast, as I said, it is a double membrane organelle with outer membrane and inner membrane. So the space limited by inner membrane is called stroma. So the inner content of the chloroplast is called stroma. And as you have seen, the coin-like structures that are piled one above the other, they are called thylakoids. The thylakoids which are piled up one above the other, they are going to form what we call it as grana. And there are also fret channels found between the granum. Right. So the number of organized flattened membranous sacs called thylakoids are present in the stroma. And thylakoids are arranged like stacks of coins, like how we pile up one coin above the other. So also the thylakoids are piled up one above the other, and hence they are going to form what? Grana. And there are flat membranous tubules called stroma lamellae, or we also use the word called fret channels, the thylakoids of the different grana. So the thylakoids enclose a space called lumen. The empty space is called lumen. And the chlorophyll pigments are located in the thylakoids. So chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. So stroma contains small circular DNA and ribosomes. Moving on to the next very important cell organelle called ribosomes. All of us know how important ribosomes are. When we differentiate a prokaryotic cell from a eukaryotic cell, one very important point is the presence or absence of ribosomes. Prokaryotic cell will not have a definite membrane-bound nucleus, as well as all the cell organelles are absent except for ribosomes. So in plant cell, in eukaryotic cell, there is a membrane-bound nucleus as well as all the cell organelles along with ribosomes. So we know ribosomes are nothing but the protein factories. They are the protein factories. Why they are called as protein factory? Because they synthesize proteins. So the process of protein synthesis, the amino acids which are picked up from the cytoplasmic pool by the message given by mRNA to tRNA, from tRNA to rRNA, from rRNA to ribosome. And the ribosome is going to arrange the specific amino acids in a particular sequence, and that to make a particular protein. Like for example, hair is a protein, nail is a protein. So each protein has a different sequence. So the arrangement of the amino acids is made possible by the ribosomes to make a particular protein. Hence it is called as the protein factory. So here you can see the beautiful picture of a ribosome having a large subunit. This is a large subunit, and you can see this is a small subunit. Right, here you can see how the mRNA and here you can see the tRNA. tRNA will collect the amino acids and it will start give to make a particular protein 20 amino acids are required. So 20 tRNAs will be there. Just like how a bus picks up the passengers, in a similar way the transfer RNA picks up the specific amino acid from the cytoplasmic pool and give it to rRNA, from rRNA to ribosome. So, but the genetic message is given from DNA to mRNA. So, as you know, mRNA is synthesized by DNA. All the types of RNAs are synthesized by DNA. So, DNA gives a command to make a particular protein. So, mRNA picks up that message from the DNA, comes out of the nucleus and guides tRNA to pick up that particular amino acid, and it gives it to mRNA, gives it to rRNA, that is ribosomal RNA, and ribosomal RNA will give it to ribosomes. So, what are ribosomes? Ribosomes are granular structures first found by George Palade in 1953. Who is the scientist who discovered these ribosomes? It was George Palade in 1953. Composed of what? Ribonucleic acid and proteins. So, they are composed of ribonucleic acid and proteins. Non-membranous cell organelles. They are non-membranous cell organelles. So, ribosomes are granular structures first observed by George Palade in 1953, composed of ribonucleic acid and proteins, non-membranous cell organelles, and eukaryotic ribosomes are of 80S, while the prokaryotic ribosomes are 70S. As I said, both the prokaryotic cell and the eukaryotic cell will have the ribosomes. So, they are characterized by the presence of ribosomes. But eukaryotic ribosomes are of 80S. S stands for Swedberg units. Then prokaryotic ribosomes are 70S. So, S stands for sedimentation coefficient measure of density and size, where we use the word called Swedberg unit. S stands for Swedberg unit. So, both 70S and 80S ribosomes consist of two subunits. Here you can see two subunits. This is one bigger subunit and this is the smaller subunit. So, primary very important function of ribosome is what? Protein synthesis. Protein synthesis. It's mainly involved in protein synthesis. Hence, it is called as what? Protein factory of the cell. So, moving on to the next very important cell organelle called cytoskeleton. So, an elaborate network of filamentous proteinaceous structures present in the cytoplasm is collectively known as cytoskeleton. How do we define the word called cytoskeleton? An elaborate network of filamentous and proteinaceous structures. Proteinaceous structures present in the cytoplasm, they are collectively called as cytoskeleton. So, what is the very important function of cytoskeleton? It is involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. So, they are mainly involved in giving mechanical support, motility, and also for the maintenance of the shape of the cell. So, what are cytoskeleton? An elaborate network of filamentous proteinaceous structures present in the cytoplasm is collectively known as what? Cytoskeleton. Cytoskeleton is involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Moving on to cilia and flagella. I think all of us might have come across cilia and flagella are hair-like outgrowths. They are hair-like outgrowths of the cell membrane. So, they are very important for what? For locomotion or for movement. So, cilia are small, help in the movement of cell or surrounding fluid, and flagella are longer and responsible for cell movement. Cilia and flagella are covered by plasma membrane, possess a number of microtubules running parallel to the long axis. Here you can see microtubules, and you can see the radial spoke here, interdoublet bridge, peripheral microtubules, and plasma membrane. So, cilia and flagella are hair-like outgrowths of the cell membrane. So, cilia are small, help in the movement of cell or surrounding fluid. Flagella are long and responsible for cell movement. Like if you come across paramecium, the whole body of the paramecium is covered over by hair-like structures called cilia. Like euglena, you might have come across the whip-like structure called flagella. So, cilia and flagella are covered by plasma membrane, possess a number of microtubules running parallel to the long axis. The axoneme usually has 9 pairs of doublets of radially arranged peripheral microtubules. So, here you can see peripherally arranged microtubules. Usually has 9 pairs of, you can make out 1, 2, 3, 4, 5, 6, 7, 8, 9. So the axoneme usually has how many? 9 pairs of doublets of radially arranged peripheral. So you can see 2 doublets and a pair of centrally located microtubules. And a pair of centrally located micro. The central tubules are connected by bridges and are also enclosed by a central sheath which is connected to one of the tubules of each peripheral doublet by radial spoke. Here you can see the radial spoke. So the central tubules are connected by bridges and are also enclosed by a central sheath which is connected to one of the tubules of each peripheral doublet by radial spoke. So the peripheral doublets are again interconnected by linkers. Here you can see the linkers. Here you can see the central sheath also, right? Central sheath which is connected by the tubules of each peripheral doublet by radial spoke. And the peripheral doublets, as I said, are interconnected by linkers. So both cilia and flagella emerge from centrioles-like structures called basal bodies. So moving on to the next structure called centrosome and centrioles. They play a very important role in the process of cell division. So centrosome, what is it? Here you can see the picture of a centrosome. It is like a wheel-like structure, right? So centrosome is an organelle usually containing 2 cylindrical structures called centrioles. Here you can see the 2, they are called as centrioles. Here you can see the 2 cylindrical structures called centrioles, right? So they are surrounded by amorphous pericentriolar materials. Both centrioles in a centrosome lie perpendicular to each other. Here you can see they are lying perpendicular to each other. They are lying perpendicular to each other. They are lying perpendicular to each other. So each centriole has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils, also called as tubulin. Here you can see the microtubulins. Right. So what are centrosome and centrioles? Centrosome is an organelle usually containing 2 cylindrical structures called centrioles. Then they are surrounded by amorphous pericentriolar materials. Both centrioles in a centrosome lie perpendicular to each other. Each centriole has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulins. Then each of the peripheral fibril is a triplet and the adjacent triplets are also linked to each other. So the central part of the centriole is called hub. Here you can see the central part of the centriole is called as what? It is called as hub, and the hub connected to peripheral triplets by the radial spoke. So here you can see the hub is connected by the radial spoke. Then the centriole forms the basal body of cilia, flagella and forms spindle fibers during cell division. Hope you have understood the concepts what I have explained in today's session. So I'll be back with the last concepts of this very interesting chapter called the cell, the unit of life. That is, we'll be studying about nucleus and the summary of all the cell organelles what we have studied so far in this chapter called cell, the unit of life. So I'll be back with some more concepts. Till then, goodbye and thank you.